In signal transduction pathways, multiple steps occur to get a specific cellular response. One kind is a G protein receptor system. The purple is a G protein linked receptor that is located through the plasma membrane. The pink is a G protein that is located on the inner side of the plasma membrane. Before the pathway starts, the G protein is in its inactive phase with just a GDP attached to it. The blue is an enzyme in its inactive state that is located through the plasma membrane. A signal molecule binds to the G protein linked receptor. This activates the G protein by trading the lower energy GDP with the higher energy GTP. The G protein then moves to activate the enzyme. The activation of this enzyme releases a cellular response. The G protein then leaves the enzyme with just GDP and releases an inorganic phosphate, returning to its inactive state. The cellular response can trigger a series of reactions using a second messenger to open the calcium-gated channels along the endoplasmic reticulum. Calcium ions are released from the endoplasmic reticulum and act as second messengers, binding to other proteins to give off a cellular response.
Unlike the G protein-coupled receptors, an individual tyrosine kinase complex is capable of activating 10 or more signal transduction pathways to generate a cellular response. Prior to the attachment of the signal molecule to the ligand bonding site, the receptor tyrosine kinase proteins exist as individual polypeptides. These tyrosine kinase receptors are made up of alpha helix within the cell membrane and have an intracellular tail composed of tyrosines located in the cytoplasm. When a signal molecule binds to the receptor, the separate polypeptides combine to form one single dimer. This joining of receptors is known as dimerization and activates the tyrosines aligning the receptor. An ATP is broken down into ADP, and the additional phosphate is added to each tyrosine on the tail. This fully activates the receptor protein. Inactive relay proteins recognize the newly activated protein and bind to their designated phosphorylated tyrosine. Once bound to the tyrosine, the relay proteins are activated and trigger a phosphorylated cascade. In the cascade, various molecules are phosphorylated through the addition of a phosphate group to the following molecule in line. The last active protein kinase phosphorylates a protein that brings about the cell's response to the signal.